And now it's time for some content again. Wim Heine, head of the Department of DNA, is going to introduce.
Okay, thank you, Ingrid. It's an honor for me to introduce Manfred Kayser as the next speaker. He obtained a PhD from Humboldt University in Berlin, and at that time he started to apply Y-chromosomal DNA analysis to the forensic practice. And he went on doing that, and he also performed research on the field of predicting physical traits from DNA, and most important things, and I think that he will be telling about it, is hair color, eye color, and some others. And he has been a full professor at Erasmus University since 2004, I think. And the Erasmus University, his group, and the group of Peter de Knijf at Leiden University and the NFI collaborate within the Netherlands in a forensic genomics consortium, and that will also be part of the talk, I think. Let me be short, because Manfred has a lot to tell. The floor is yours, Manfred.
Thank you for that kind invitation, and I do thank the organizers to be able to speak to you here today, and in particular to be able to speak to so many of you today. So I do realize that not all of you have a strong DNA background, so although you may see some details in the slides that may not be too familiar for you, I do try to explain in a more late-term language. Yeah. The idea actually is with this talk, and I should start with a disclaimer because I only talk about human stuff. Yeah, so there's lots of other advantages going on in forensic molecular biology that do not concern human but other creatures. That is not in this talk. And I should, of course, mention that there's a strong bias here. Because what I see as advances and progress usually is things we are doing in Rotterdam, and I hope that at the end of the talk at least some of you will agree that indeed these are the mainstream new challenges, although it's by far not complete, and so you may miss some aspects. So the first example I want to give to you where currently there is quite some exciting progress is in the field of Y chromosome analysis. You may know that YSTRs with commercial kits are used since a couple of years for male identification, but the disclaimer here is it doesn't really work on the individual level because close male relatives have the same Y chromosome. Why is that? Because only males have Y chromosomes, and you basically get the same Y chromosome from your father because there is no what people call recombination. And that is, of course, a disadvantage. Because you cannot draw conclusions based on the individual level. So for a long time that has been considered as, well, that's what we can do and we can't do much better. But there may be a solution actually which we followed up, and it looks quite promising, not in every case, but in quite a number of cases where indeed you can differentiate between male relatives. And how can you do that? Well, it has to do with the natural rate of change of those STRs on the Y chromosome. So what we therefore did is we tested a large number of markers in 2,000 father-son pairs where we knew it's father-son pairs because we had DNA evidence that they are. And this included actually all the ones that are in the current kits. And we simply asked how often do they mutate? So how often do they naturally change between a father-son pair? And that's actually what you see here. So these are ordered according to their rate of change, and the rate of change you see here. So what are these numbers? Well, basically 1 times 10 power to the minus to the power to 2 means that there is 1 mutation every 100 father-son pairs. As you see, most of these YSTRs are way below. So there was no mutation in these 2,000 father-son pairs, and there are some where the mutation rate is about 1 in 1,000 or a few in 1,000, which is still fairly low. And I'm not sure if you can see that here, but these green spots are the Y-filer markers that are included in the commercial kit. So they're all fairly slowly mutating. But then at this point here you see these red dots, and you can see a little jump here. And that illustrates that there are some of these STRs that have a much higher mutation rate. So now the advantage with those is that obviously you can use them to differentiate male relatives. And you can describe that theoretically based on the mutation rate, but you can also simply look at male relatives and simply ask how many I can differentiate. And what you see here is male relatives, a couple of hundred, that are separate by 1, 2, 3, 4, 5 male generations. Yeah. So father-son is 1, brothers is 2. And in gray, you see the values you get with Y-filer. So you can't really differentiate many of the close relatives with Y-filer, but in black you see these 13 rapidly mutating YSTRs, and as you can see here in this sample set, which admittedly is still small, but we're working on getting it larger, about half of the father-son pairs we can differentiate, about 60% of the brothers, and so on. And obviously, if you increase the number of generation male relatives are different, you will also find, or you expect more chance for mutations, and therefore these numbers go up here. So on average in this set here, we could differentiate 66% of these male relatives that come from all different levels of relationship, and that compares with only 15% with the commercially available YSTRs. And the reason simply is that we're dealing with markers that have a much higher mutation rate. By the way, those markers are also very useful just to differentiate groups of male relatives, so-called male lineages. Again, in gray, the Y-filer markers. You see that in a global set of these worldwide individuals, you just get a bit over 0.9, where 1 would be the maximum you could reach, because these are all supposed to be unrelated people, which is a bit of an issue in these samples, but almost. And with these RM YSTRs, you reach values that are well above 0.95. Actually, you only miss 1.7% of these individuals, and some of them actually are related in a 3rd or 4th degree. So where does that lead to? Well, we currently, and perhaps some of you are working with us in this project, trying to collect population data for these rapidly mutating YSTRs. There are 70 labs worldwide that are involved. We are producing currently 10,000 individual data sets, which we try to make available for future forensic application. There are discussions with at least one biotech company that is quite involved in forensic analyses to make those commercial. And for those that are really in this business, you may have seen that Promega has a new YSTR kit. So this kit has 6 additional markers compared to Y-filer, and 2 of them, not by chance because we published on them earlier, are belonging to those rapidly mutating ones, but the other 4 do not. Okay, so let's shift gears now, because YSTR, and in particular using rapidly mutating YSTRs, is still something that allows you to identify people. But as with all current forensic DNA analysis, it only allows you to identify someone you already know, because it is a comparative analysis. So you have to have the suspect, because you have to have the DNA profile of the suspect, which you may then want to compare with the DNA profile of the crime scene. If you don't have the right suspect, or every time you try to match the profile with the database, it doesn't match, that's it. That's it for the use of DNA in forensics. And obviously there are ways out of that. One is DNA mass screenings, which in some countries are applied, usually not so often because of lots of issues, ethical, financial, other issues. The easiest way, obviously, is to enlarge your criminal DNA database, which usually is not in the hands of the forensic scientist, because it should be a societal and political decision. So out of that, the idea emerged that what if we can do further and use DNA in a more investigative approach? So what if we could predict or describe from the crime scene sample the appearance of the person who has left the sample? And this is what some people call forensic DNA phenotyping, where currently the phenotype actually is visible traits, its appearance is not disease and other things. And the idea is very simple, because if you can describe this, and the more exactly you can describe this, the more the better it works. Obviously, out of a large number of potential suspects, which could be very large if the police has no idea where to look for, you can actually lower this number by describing at least a group of people that match the appearance criteria you got from the crime scene sample. And by that, you can concentrate police investigation. So this is not actually omitting STR profiling, but it makes it more effective, because you may want to do this not on thousands of samples, but just on perhaps the 100 or even less that match the appearance criteria you get from the crime scene sample. So I'll show you a few examples how that works, and the best example to illustrate that is eye color. Perhaps one can dim the light a little bit because these are a bit light-sensitive slides. So I hope you see that, as you know, there is eye color variation in the human population, but only when it concerns Europeans or people of partial European descent. So at a time, we were interested in producing the best markers that can actually predict eye color. And what this is here is something what medical people use to predict your and other people's disease risk from DNA data. So this actually is a population average prediction value that runs from 0.5, which means like tossing a coin, you don't use any of the DNA information, to 1, which means every time you predict correctly. And what you see here is you accumulate genetic markers. These are not STRs, these are SNPs. And you do this for a while and you see the saturation effect. But what you see overall with these bunch of genetic markers is that you reach for blue and brown eye color a value that is well above 0.9, well, not well above here, but in other studies we have done, where 1 is completely accurate. So it's quite predictive, actually, and obviously it doesn't work as good for the non-blue-brown eye colors, which you may imagine. It doesn't, because this is actually not the extremes of the distribution as blue and brown are. So what we then is, we took a cutoff, say 6 genetic markers from 6 different genes, and we developed an assay. We published on that, and we developed a model where, based on thousands of data where we have eye color and genotypes, you can actually estimate the probability, given a certain genotype combination, this person rather has blue or brown eye color or intermediate eye color. And here you see the genotype combination that gives you high frequency for brown. This is the combination for high frequency of blue. So how does that work in practice? And obviously one cannot dim the light, otherwise or people are not hearing me. This is actually an example of 40 people from the department or across the floor, and those eyes are not ordered as you would order them by looking into the people's eyes. They are ordered as these 6 SNPs in this prediction model would predict different probabilities for blue, brown, and intermediate, starting with the highest probability for blue here, and then you go down, highest probability for brown here, and then you go this way. And if you would have all the time you need to understand that, which unfortunately I cannot give you, you would see that there are 3 people here out of those 40 where actually in 2 cases you have at least 2 similar probabilities. So you would have to conclude that, well, this is probably not very informative in this individual. And there's 1 where you have quite a high probability for brown, but the eyes are not brown. Yeah? And this gives you the prediction accuracy of above 90% we would expect from the population-based analysis, but on an individual-based analysis. So therefore, eye color is an example where actually we managed to go through the entire system. So we found genes, we developed predictive DNA markers from those genes, we developed a test, and we evaluated the test. So this is SWIGDAM validated, this array, and we also validated the prediction model, not only in the thousands of Dutch Europeans we used to describe the model, but also in thousands of Europeans from other areas, and actually the prediction model behaved very similar. So it's all there, it's all published, you can all use it, and the Dutch lawmakers took that knowledge in allowing DNA-based eye color testing in the Netherlands since April 1st this year. So everything else is more disappointing, currently, I should say. Perhaps hair color is the exception, and Susan Walsh will tell you more about our work on hair and eye color prediction in practice, in particular in a talk on Thursday morning. And we are just having a discussion with Wim, so it looks that the paper describing this high-risk plex test we have developed with all the data seems to be out today, so there may even be some press attention. So what we actually did last year is doing this population-based study by looking for the most informative hair color markers. And as you see here, with 22 DNA variants, we are reaching these values that would be 1 if completely accurate prediction would be obtained. They are not 1. They are quite above 0.9 for red hair, which is not a surprise because it's 1 gene majorly involved. They are a bit lower for other hair colors, and they are lowest for blonde. And Susan will tell you more about that if you're interested, but I'm a living example why currently we can least accurately predict blonde, because I was white blonde as a kid. I probably have the genes to be white blonde. I'm not white blonde anymore. Why? Because there's age-dependent hair color change with some people, and nobody yet has any idea what the molecular basis of that is. And therefore, it's not yet in those models, but we are working on that. So just to give you a glimpse of what else is around there. So you may think, well, body height must be easy to predict. And indeed, if you ask twin studies, you get about 80% of heritability for body height. So 80% of height variation in the human population should be in the genes. So we once did a prediction studies after the first papers came out. And this is the value that is 1 if it's completely accurate, and it's 0.5, like tossing a coin. So you use 50 SNPs that at the time had been highly statistically robustly associated with height, but they hardly predict height in an accurate way. So why is that? This is a study we did where we looked for more height genes, and this is the number of individuals we were using to find the first new gene after the first genes have been published already. And the problem is here, actually, because for this SNP here, which gives a nicely robust, as the convention agrees, statistical association with body height, you get a 0.4 centimeter, namely 4 millimeter increase, decrease if you have to have that allele in whatever homozygote form. Yeah, so the individual SNP effect on height per each gene or marker is tiny. And that explains why even in a large study, and that's the largest genetic hunt for body height genes ever published so far, where 180,000 people have been investigated genome-wide, 180 genes have been found. What do they explain in these 180 people? Just 10% of their height variation, where the assumption is 80% should be in the genes, so we are still missing 70%. And that's the reason why we are still out there to look for more height predicting genes, because currently it doesn't really work. One other example where you may think, well, this is hardly to predict because it should be so complex, is age. And obviously age is a visible thing, not really with point age, but usually people can differentiate if someone is whatever 20 or 40 or 60 or 80 just by looking into a person. So we do see age more as a visible trait. And the question is, can we predict that? Well, we developed a test that actually quantifies the number of thymus cells. You may know from biology back at the time that the thymus is an organ which actually goes away with increased age. It doesn't go away, but these thymus T cells get replaced by fat cells. So we lose T cells all the way we age. And that's actually what we took advantage of. So this is a normalized quantification DNA test. And the value we get actually has a very strong relationship with age, as you see here. So it's quite a strong correlation coefficient. It comes with a standard error, as you may see here. So it's not really useful yet for individual-based or for point age estimation, but if you group the individuals according to age groups, and these are more the visible age groups, 20, 40, 60, 80, you indeed reach high prevalence-adjusted prediction accuracy. So there is something in there, at least for group age estimation, that's quite useful. And indeed, we are working on making this more efficient. We have developed some other biomarkers, and we could actually get the correlation higher and the standard errors lower. And this is actually the development we are continuing. So where does that lead to with appearance prediction? Well, we need more predictive markers for a couple of traits. Basically, all of the traits you have seen here, including eye color, because remember, we can nicely predict the blue and browns, but we cannot predict the greens or the light hazels or the yellowish ones. They are all very difficult because they all fall in this intermediate group. And what else is out there? There's a few genes out there for, say, hair morphology, right? If your hair is curly or straight. There's one gene that has been found. Baldness, some genes have been found. It's not by chance a male phenomenon because the gene with the strongest effect is on the X chromosome, where poor men only have one. And if you have the wrong one, then unfortunately you can't change it, right? Facial shape, well, until very recently I would have said there's nothing there. There was one paper published earlier this year, and there will be one paper, actually is our paper, published early September in PLOS Genetics, where we did thousands of individuals. We took images, we looked for the genes. This, not by chance, should resemble me, because it's me from an MRI image, which we actually used to find the genes. And this is just for those that are into gene mapping. So we find 5 genes, actually, that we believe are involved in facial shape. So it's much too early to use this for any prediction analysis, but people now start to understand what genes are involved in the human face. And obviously, if you think about it, there should be genes involved. Why? Because we know that monozygotic twins are almost identical in their face, and dizygotic twins, which are just siblings, are similar, and they're usually more similar than unrelated people. And that tells you usually, yes, it must be in the genes, but how complex it is, we don't yet know. We're just starting to understand the genetic basis of the human face. So with that, I would like to lead to a related but not really the same issue. And usually people try to mix this up in some ways that cause quite some scientific hiccups. So it's the same rationale. You are led with a problem that you cannot identify based on DNA profiling people you do not know. In other words, you don't have a DNA profile. But what you try to do is a bit different, because you don't try to predict appearance, and that is simply because appearance prediction is just around from a scientific basis since the last, say, 5 years. But what was around much longer is trying to understand the relationship between your DNA and where you and your ancestors come from. So biogeographic ancestry or genetic ancestry or whatever continental ancestry you may want to call this, the scientific evidence for that is around much longer, and therefore no wonder that even in the forensic field people got interested in that. So usually what people do is they use the Y chromosome, not only because usually perpetrators are males, but also, as you see here, every color is a different type of Y chromosome, genetically characterized. There's quite some variation here, and the distribution of these types is not random. So you see certain types of Y chromosome that are predominant in certain geographic regions. And obviously this can tell you a lot where at least the paternal ancestors of that individual came from. And you can do the same trick for mitochondrial DNA, which obviously males also have, but they don't get from their fathers because all human mtDNA is transmitted via the mothers. And again, different types of mtDNA, different major mtDNA types in different parts of the world, and therefore quite useful. The problem actually is if you by chance, and in forensics now you never know, hit a person where the father and the mother came from different continental regions. And then of course you get conflicting answers, and you wouldn't necessarily know what to tell the police they should be looking for. So therefore we and others went to markers which you get from both of your parents. The problem is that the amount of geographic information at those markers by nature is much, much smaller, and that has to do with certain genetic effects. So what you see here is an analysis based on almost 650,000 genetic markers scattered across the entire genome in about 1,000 individuals across the world. And what you can actually see, if you actually use these big numbers, you can separate people according to their continental origins. You see the Africans here, you see the East Asians there, Native Americans here, Europeans there, Middle East there, Central Asia. There is some overlap for some groups and some individuals, but usually you can get quite some good information about the continental origin out of that. The bad thing is that usually these markers come on little SNP arrays, glass slides. They usually don't work in forensic scenario because they like to have more DNA, they like to have intact DNA. So therefore people like us and others try to find the most informative in respect of geography of the 100 and 1,000 and millions of those markers that are out there in the genome. And here you see the same individuals now being analyzed at 47 so-called ancestry informative DNA markers. And obviously you lose resolution, yeah, so same colors. The separation is not as strong anymore, but at least it allows you to differentiate most of the Africans from most of the East Asians, from most of the Native Americans, but then you are left with a big group of what actually is Eurasia. Europe, West Asia, Middle East. And if you think about that, obviously this makes sense because there are lots of migration movements between the Middle East and Europe and West Asia and Europe, and they don't allow stronger genetic differences to create. But obviously, if you increase geographic distance, then you expect a bit of a larger genetic differentiation. But obviously, you can develop tools based on that smaller number of SNPs that can be applied to forensic DNA analysis simply because you can use more appropriate technologies. So we, together with Peter, for instance, with an FGCN, Peter de Knijf, we were interested in finding those most informative ancestry marker sets. So here you see a picture of only 10 SNPs, 10 genetic markers only, and the picture is very similar as with these 50. So you can differentiate Americans from East Asians, from Africans, and then left with a big group of Eurasians. If you play around with that a little bit, you add another 10. You do see that the Europeans become a bit different, but usually it is not so easy to differentiate Europe from their direct geographic neighbors, at least based on these autosomal markers. So can you stretch that further? Can you say something within a continent? And again, until a few years ago, people would have said, no way. And it's still more or less no way, but there are pictures that emerge. So what you see here is a genetic map of Europe, where 2,500 individuals were used, large number of genetic markers, again, those SNP arrays. And this genetic map is quite highly correlated with the geographic map of the sampling sites of these 2,500 individuals. Obviously, you may not even be able to read that here, but just as a take-home message, every of these signs here is a single individual, and you do see that those similar signs are clustering together, which basically means that they are more similar to their sampling neighbors than not. But there's also geographic information here. So this actually is all the Northern Europeans, this is all the Western Europeans, this is the Southern Europeans, this is the Eastern Europeans, and these are the Finns, who are known to be genetic outliers as also showing up in that analysis. And then with the black dots, actually, you see here samples that are often used as representing Europeans in genetic analyses. So these are European Americans living in Utah, where the family records would tell that they come from Northern and Western European countries. And all, perhaps except one, actually are matching in this map with other people from Northern and Western European countries. So you can even do it a bit further, and using a method which we are currently developing, which is very sensitive to even detect small genetic differences. And you can see you can group Europeans in 1, 2, 3, 4 geographic groups, and again, the Finns appear as outliers. So with this, I stop with DNA. I hope I have informed you a little bit that indeed there is more out there to use DNA for investigative purposes. But there are other developments out there that actually may help you once you have identified a DNA donor to make the link with the criminal act, which usually is not the task of the forensic scientist, but the task of a judge. Yeah? But there may be information in your crime scene sample that allows you to give information that at least excludes certain scenarios. And one of the links here would be cellular origin. So knowing from what type of cells you have extracted your DNA profile. Because this actually can be quite informative. So think about a scenario where a stain ends up on a blue dress, and I'm not giving names here, right? And the question is, what is the cellular origin? And I guess the person who left that stain there would have had less problems to explain this if you find out it's a saliva stain than it may be a sperm stain, right? So these type of scenarios obviously exist in practice and may be useful. So obviously there are helpful aspects. There are protein-based tests, there are chemical-based tests. Usually they are not very accurate. They are quite accurate, therefore they are used in forensic practice. I guess you're using them. But there are scenarios where they give you the wrong result. So the question was, can we develop more specific answers based on biomarkers? And this is a field that has been booming in the forensic genetic arena for the last 4 years or so, and perhaps many of you are involved. So just to give you a bit of an idea how that works, so this does not work based on DNA, it works on RNA, which is the next step, yeah? And actually it works on the quantification of RNA, which people call gene expression. So it basically is a matter of how active a gene is or not. And there are strong differences actually between tissues. So if I do a genome-wide analysis on this gene expression and I kick in a couple of individuals where I had blood, semen and saliva, this analysis does not order these samples according to individuals, it orders them according to body fluid. Yeah? And that means the individual variation actually is much smaller than the variation between body fluids. And from that rationale and resource, people have picked quite some informative markers, like you see here for blood, where you see the high bar means there's lots of this particular RNA there because the gene is very active, and the low bar means there's not, comparing blood and saliva. And here you see strong differences between blood and saliva for a number of genes and a number of individuals. So as you can imagine, people have picked quite some informative markers. Usually when they compare forensic tissues, you get a certain answer. You may still want to look into other tissues, although you may not encounter them that often in forensic casework. But as in this case, you do see here, if you compare like 62 body tissues from the human body, these genes only appear in those that are involved in blood. And that is because there are specific things going on in blood in terms of gene expression. So those of you that are remembering RNA from school, they may have that thing in mind that RNA is very unstable. Yeah? And indeed, many RNA molecules are much less stable than DNA on average. But if you take this into account in your search, as we did, because we used degraded samples, you actually find markers that even after 16 years, at least blood stains that were kept in the desk drawer, you can still detect those genes. So there are RNA markers out there that indeed are very stable. People have developed those markers and even multiplex tests now. The NFI was involved in one of those for all types of, actually for nearly all forensic relevant body fluids. This is something we did lately for skin, and that's quite important if you think about trace DNA. Right, so you may want to differentiate if your DNA profile really comes from someone having touched an object, or if he, as perhaps I now have, a bit of a wet speech, and therefore it's saliva transmission, because there are different invasiveness involved in those type of scenarios. And indeed, you can have RNA markers that show a strong difference between skin, like palm, like touched object cells, and all types of other forensically relevant body fluids. So the other type of information you may get from a crime scene sample that actually does allow you to link or not the crime scene sample donor with the criminal act is time. And obviously, many of you have thought about that already. If you would be able to say when the stain has been placed at the crime scene, at least you can use that information for alibi testing. And there's no current solution. There are actually two aspects. I will only explain you one of them. The one I don't explain, there is some knowledge there. I saw a poster that describes this based on the one system that currently is out there, and that actually indeed gives you an idea about how long the sample is sitting at a certain place in terms of days, weeks, and months, so which actually is the age of a sample. What I would like to show you is another aspect of trace time estimation, and that actually answers the question when within the 24 hours day-night time the sample was placed at the crime scene. And why can you do this? Well, you can relate forensics to chronobiology, because we all know that there are some biomarkers in our body that are very much dependent on day and night time. And those of you that came from overseas to these beautiful countries, they are actually suffering from that, because you're suffering from jet lag, which is caused by this melatonin hormone, which has a strong peak intensity at the late night, and during daytime is low. And actually this is the hormone that allows you to sleep. And the American colleagues may not care because they have melatonin pills with them, and therefore can reset their biological clock. But usually this is what you're suffering if you suffer from jet lag. And there's another one, for instance, here, cortisol, where the peak is a bit shifted. Yeah? And these are the markers, of course, you want to have. So a marker that changes concentrations quite a lot within the 24 hours. These are called circadian biomarkers. And what we did in a kind of proof of principle study is actually trying to capture that circadian signature from small amounts of blood and blood stains, which is not so trivial because usually these tests work on plasma or larger amounts of blood. But as you see here, this is exactly the peak time for melatonin you expect, about 3 a.m., flat, no concentration during the day, and cortisol, there is some background concentration, but in these there's a big peak more in the early or later morning. And obviously, if you would combine these two biomarkers, and obviously we are trying to find more, at least it allows you to give a 4 or 5 hour range during the day and night, simply based on these two markers. Obviously, what you have to show and you have to make sure is that once the blood has been donated somewhere, this signature doesn't change anymore. And if your hormone would be started to get degraded, then it would change because you would lose signal, you would lose concentration, and by that you would do the wrong timing. And we tested for that, at least preliminary. So samples between fresh and 28 days. And as you see here for melatonin, different individuals, there's almost no change. So this melatonin hormone concentration stays stable over sample storage time. And there seems to be a bit of a decay here for cortisol that may be more difficult in the longer storage times. So we're working on trying to improve this, and you see some examples of genes now based on expression signals, mRNA, which have very different behaviors within the 24 hours, and by that we hope that we can actually narrow down the window within the 24 hours, you can say, by circadian markers, for instance, from crime scene analysis. So last example I want to show you is, and that is a bit futuristic, I admit, what about other things you may want to extract from the crime scene sample? Because if you think about it, there may be certain biological conditions, say physiological conditions, at the time of the criminal act, either in the perpetrator or in the victim, that actually may help you if you are able to find that out from the crime scene sample in detailed crime scene reconstruction. So obviously this is nothing we can do at the moment. But at least theoretically, you would obviously expect that many of the things that have forensic relevance, think about fear or angst, right? So if the victim sees the perpetrator and obviously seeing the weapon and getting frightened, I mean, this is a physiological condition. And in principle, you should be able to find the biomarker, neurotransmitter or something that causes this, and then perhaps you can use this as a diagnostic tool. So nothing is out there yet, but just to illustrate that, I mean, physiological conditions can be wide and not criminally related, but sometimes with a relevance for crimes investigation. This was a case where the NFI approached us actually doing a simple pregnancy test from an old blood stain, where you can't actually use those nice pharmacy-based tests, which you usually use at home with urine. They don't work on an older blood stain. So what we developed actually is a genetic assay, again based on RNA and gene expression, because there are some genes which actually are expressed in the placenta, which obviously you don't have if you're not pregnant, and by that you can differentiate pregnant from non-pregnant women, which may provide, at least in this case, I think it was relevant, some investigative leads. But these are the kind of things that may be ongoing. And I'm not saying that this should be the future, but you can even do this one step further, and this is not actually asking about the physiological condition, but asking about the genetic predisposition of a person. Yeah? So what is the risk of a person, for instance, having or to develop a certain behavioral trait? And of course, with forensics, the big thing is in aggression, which some people call impulsive behavior. So instead of talking with your neighbor, as nicely enough, that's a typical Dutch trait, you start hitting your neighbor because you can't stand all the talking anymore, right? So there is something in the genes, and I'm not saying that this is the way it should go, it actually had been applied in front of court. And you may have seen that news headline. So there's one particular gene which scientifically indeed is associated with impulsive behavior. And there was one case in Italy where the genotyping and the result of a certain perpetrator that was shown to have this more violent risk allele and gene was actually used in lowering the sentence of that person. This is just to illustrate to you where it could go. Obviously, it should only go this way if the scientific knowledge we have is very, very solid. And at least currently it looks that the knowledge we are developing for externally visible traits at least is more accurate and more reliable so far than our understanding about behavioral traits. And with that, I'm finishing, and you may have started wondering, well, this is all coming from your group. Who is working in your group? And I have to admit that there are more people than are usually working in the forensic department, because we really have experts from very different fields, which obviously you all need if you want to really go from a fundamental science to a more applied science. So I do appreciate that these people are all with me and doing those exciting research projects. Obviously, you also need collaborators that are very happy to do the work with you. And here you see a number listed. And you need the money, and these are the funding agencies, in particular the NFI, which is funding our work for a number of years already. Thank you very much.